Shiv has a very interesting career trajectory. I've got his CV here. So I started off in Pune in India with a bachelor's in mechanical engineering, where I note he was ranked in the top 3 in your class, a very big class and a big school. So really destined for great things as is laid out in the CV. So then a master's in at UIUC, also in mechanical engineering, before 2 years off working in a company doing kind of fluid dynamics modeling for biomedical devices. And then returning to academia, this time with a bit more of a biomedical focus at Northwestern, and then followed by a really successful postdoctoral period with the famous James Sputtage at Stanford. I think there's over 10 high-profile papers listed from that time here in the CV. And then joined the faculty here in 2011, primarily cell and developmental biology. Shiv got a NIH New Innovator Award very recently and is doing very well on the grant side. And it's a pleasure to have him here because he's very supportive of younger scientists, and my interactions with him to date have been very positive, and he's very supportive of more junior faculty as well. So thanks for accepting the invitation, Shiv.
Can you guys hear me in the back? Yeah. So kind introduction. Thanks, Patty, for the invitation to give a talk, and look forward to sort of interacting more with DCM and B, and maybe forming some collaborations and doing some work together. We're right across the street. So we like to call ourselves the Protein Acrobatics Lab. Hopefully that'll become apparent as the talks go along as to why we do that. And the title of my talk is sort of trying to stem this bridge between protein interactions and cellular function. And to put that in context, a cell type that we're interested in working on is the cardiomyocyte. So this is basically a single cell that can recapitulate all of the functional properties of the human heart. So it brings about contraction. It can respond to hormones. It can sort of adapt to different types of stress that are put on the heart, whether they be physiological stressors or pathophysiological stressors. So it's a really nice unit of the heart in which to study the properties of the heart as a whole. And the challenge is that in disease, you see large changes in the structure and function of the myocyte. So you see this is a myocyte from a donor heart, normal donor heart, and this is from a failing adult heart. And you can see the myocyte is enlarged, and that is hypertrophy. It also happens when you have hypertension. You also see diminished contractility. So in hearts that are, if you look at the force production by the myocyte, in sort of a force time curve, you see that in a failing heart, you have reduced force being produced by the cardiac myocyte. And also, when you start looking at the organization of myocytes in the heart, you can start seeing disarray in terms of how the myocytes are organized, so at the tissue level, but you can also see that in terms of the actual packing of the individual protein components that make up the myocyte. So here you have thick filament distance, which is the distance between the contractile units. And as hypertrophy progresses, you have a gradual unraveling of the myocyte. And we're interested in sort of understanding how these disease conditions emerge from changes in the signaling networks that lead to these conditions. And as is common, once you start approaching signaling networks, you have an interplay between multiple signaling pathways. So here's now a dated review from Jeff Mulkenton, where they have mapped out some of the signaling pathways and how they intersect in the context of heart failure. Of course, I'd say heart failure or cardiac disease, but I could replace this with pretty much any disease condition, say cancer, neurodegenerative disease, and you would have sort of a complex map of components in the signaling pathway. And what we're really interested in is trying to understand what are these subtle rewiring events that happen in this network that contribute to disease. So when you look at a signaling network, at the heart of it is a series of protein interactions that communicate information, say from the outside of the cell and through the cellular matrix. And so here's an example of a cardiac myocyte that is being signaled to undergo hypertrophy. You have a receptor on the surface of the cell. This is the angiotensin II type 1 receptor. You activate it with angiotensin. This relays information to GQ. GQ then causes an increase in calcium and inositol phosphate, activates PKC. PKC goes on to activate ERK1/2. That goes into the nucleus and then initiates a fetal gene reprogramming event that essentially then causes the myocyte to increase in size. And so we're all used to seeing these sort of stick diagrams in reviews and at the ends of papers. The problem is that they're often misleading in the sense that there's multiple layers of complexity at each stage of the signaling pathway. And by that I mean, first, you have 4 layers, and I'm going to describe them. The first one is conformations. So the same receptor, angiotensin 2 type 1 receptor, it responds to angiotensin, but it can also be activated in response to mechanical stretch. So if you have hypertension, or let's say you have physiological load on the heart, so you're exercising, you could have activation of this receptor. There's an analog of angiotensin which also activates this receptor. And people have used structural studies to show that the conformations that the receptor adopts at the molecular level are distinct for distinct inputs. And these then translate into differential selection. And it's important to recognize differential selection in the sense that angiotensin is more likely to activate GQ. It could also activate arrestin. This ligand activates preferentially arrestin, and then both of them can activate GI as well. And it's not just an exclusive one-to-one pairing, but it's sort of differential. And that balance of signaling that's initiated through these interactions then gives rise to sort of different subtypes of how the cell behaves. Another example of this in the context of heart failure is the beta-2 receptor, a receptor we study often. And beta-2 normally is a GS-coupled receptor. So in response to catecholamines, you'll get an increase in your heart rate. But in heart failure, multiple groups have suggested this is somewhat controversial, that you then can also get activation of GI. And instead of hitting the accelerator, that's like hitting the brake. And so despite the fact that your heart is being flooded with catecholamines, it's not responsive. That's at least one hypothesis in the field. And people think that this is possibly caused by differential conformations. So the one layer above that, once you go into the cell, is multi-domain proteins. So here you have a one-to-one pairing and differential selection. If you go into a protein like PKC alpha, one target of PKC alpha is ERK1/2, but it can also go to the junctions where it can phosphorylate connexin 43. It can go and activate phospholamban in the sarcoplasmic reticulum, and it sort of has to differentially modulate these effectors. And one way it does that is by virtue of the fact that there's information in these multiple domains that allow the protein to sort of differentially select between the stimuli. So you've got conformation, and you've got selection, and you've got multi-domain, and ultimately you've got multi-protein. So in multi-protein, the best example in the cardiac myocyte is that movement in the cardiac myocyte comes from interactions of multiple myosins with actin filaments. And all of these have to mechanically coordinate their interactions so as to give rise to contraction. So you see sort of this multi-leveled, in each protein you have these, as well as in each set of interactions as you span up, you have these layers of information that we have to sort of factor in when we try and understand what's different between, say, normal and disease. So my lab is sort of interested in tackling this problem, and I'll tell you about 4 sort of vignettes of projects from our lab that highlight how we're trying to study this problem. So I'll talk about conformation, selection, multi-domain, and multi-protein. So for conformation, I introduced the beta-2 receptor that potentially differentially signals through GS and GI. So the canonical view of the G protein coupled receptor is that of a rheostat. So basically you have a single protein, you have ligand that drives the conformation, puts it into the on state. Depending upon the amount of ligand, you get variable levels of activation of the G protein, which then goes on to activate a second messenger cascade, resulting in, say, amplification of the signal. Now, what happens though is when you look at this in a physiological context, if you throw a hormone on a cell, you not only see one second messenger response, but you typically see lots of different things happen. So a classic example is you would get cyclic AMP, you might also measure calcium changes, you might also get ERK1/2. And the challenge is, well, is it an upstream or a downstream phenomenon? In other words, cyclic AMP can activate pKa, can go to ERK1/2, but this receptor could also activate, say, GI or arrestin, and that can go to ERK1/2. And this is a common problem in signaling cascades, is sort of putting components and linking them together. It's not difficult to identify things that are changed. It's connecting them that's really the problem. So what is now generally accepted from the perspective of the GPCR is that it's not a simple on-off switch, but it has more of a dynamic conformation landscape. So here's a study from Baron Kobilka's group showing that, okay, you have an inactive state of the receptor, then you have an active state of the receptor. And if you add a ligand, say in this case an agonist, you stabilize a variable conformational landscape, and the receptor can populate these conformations. And let's say in the presence of G protein represented by nanobody, you can have one conformation potentially preferentially stabilized. What that says is that what is available to bind to the receptor can also determine the signal. That's sort of a model or a theoretical model. There's a lot of supporting evidence that you have these multiple conformations. So people have, say Martin Lose's group has done FRET-based sensors. Here's an alpha-2 sensor, but basically norepinephrine would cause a decrease in FRET, so increase in CFP, reduction in YFP. A different ligand would essentially cause the reverse profile. So two different ligands in a FRET sensor can show two different changes. Here's other examples using F19NMR from Kurt Wottrick's group, where you can profile different ligands depending upon the conformations that they stabilize. And here's an extreme example from Bob Lefkowitz's group, where they take a series of beta blockers and show that the propensity to label a cysteine at the base of the receptor, which is sensitive to the conformation of the receptor, is very variable across different ligands that they essentially look for. This is ligand accessibility. The difficulty with taking this data and then going into a cell is that yes, there are multiple conformations, but how do you translate this conformation into function? And ultimately, that information has to be relayed through a protein interaction. So here's the GPCR G protein interface from the Kobilka-Sunohara structure. And it's been known for a while that you have this helix on the G-alpha protein that essentially acts like a flag. It inserts itself into this battle of the receptor, and that's shown in here, zoomed in here. And if you were to replace this helix of one G-alpha subunit with the helix of another G-alpha subunit, some receptors will essentially switch where they signal. In other words, this is one element that can be used to sort of detect a conformation, not just in terms of differences in conformation, but also have some information about what the selection looks like. So we decided to use this information to sort of build a GPCR G protein interaction sensor. We take the full receptor and we drop in this element that I'll talk about in a moment, which has a FRET acceptor, a FRET donor, and this unusual structural element in between. Then this is the C terminus of the G-alpha subunit. So essentially, if the receptor populates conformations that favor binding of this G-alpha C terminus, you would lock the receptor more frequently in this state, the system in this state, and you would get an increase in FRET. And so this element becomes then important because you have to space the two donor and acceptor such that you have minimal FRET in the basal state, then you have high FRET in the activated state. And the element that we use that sort of allows us to make these sort of interaction sensors is an ERK linker. And it's essentially a single genetically coded alpha helix. And you can put it between any two proteins. And what we found using a range of structural and biophysical studies is that what it does is that it populates sort of an extended conformation. Here's sort of a Monte Carlo simulation showing conformations that this helix populates in solution. And it populates sort of an extended conformation. There are breaks in the helix that are stochastic, which then control the on rate of the interaction. And the off rate then, the proteins, once they interact, they just come off of the native off rate. So this is genetically encoded, so all of this is encoded by a single polypeptide. And what's nice about the system is that you can vary the length of the linker, and you can vary very nicely the effective concentration that the two proteins see for each other in that interaction. So regardless of the biomolecular dissociation constant, you can modulate that interaction in a cellular environment by varying the length of the helix. And what's also nice is that you can start at concentrations of about 10 micromolar, go down to concentrations of about 100 nanomolar, and that's sort of a nice range in which to be to modulate weak interactions that are sort of classic of, say, signaling interactions that are happening as opposed to strong or very weak. So we decided to use this technique. I'll just show some quick snapshots of data for the receptor sensor. So we have the receptor with this element in between, and either no peptide, or in this case, a peptide derived from the G alpha C terminus. So S-pep refers to the peptide from the G alpha S C terminus. This is just showing that the sensors are intact, expressed at the right molecular weight. Basically, there's not much degradation of the protein. Protein nicely accumulates in the plasma membrane, which is what you would expect for a receptor. Protein is functional. So the beta-2 receptor has basal activity. So when you express it, you get increase in cyclic AMP levels. If you're stimulated with a canonical agonist, isoproterenol, you get, again, an enhancement in the signaling. And also, you can block that by using an inverse agonist. All of this is just to make the point that even though this has got all these elements tethered at the tail of this protein, it looks, for all practical purposes, like an endogenous receptor would. Now, when we start mapping these interactions, when you throw isoproterenol, which is a canonical agonist, you basically see an enhancement in the interaction for the S-pep, but not for the peptide from transducin, I, or Q. So it's really specific. Also, this is all being done in live cells without lysing anything, just making measurements using a fluidometer. And you can then monitor you can come up with sort of dosing curves where you can look at increasing concentrations of isoproterenol. You can see this nice increase in FRET. This is now mimicking the low affinity bound state of the ligand. ICI, in this case, is an inverse agonist, binds at high affinity, so it competes off the interaction pretty effectively. So now that we have this characterized system, we can go back to that question of conformation that I posited earlier, which is it possible to detect using the system an S-based conformation versus an I-based conformation? And untangling this is an important problem because S and I both act on a common effector inside cells. So GS is like the accelerator in the cell, GI is sort of the brake. So if you try and measure this level, you don't know if essentially you're reducing this or you're increasing that. So taking a foot off the accelerator or pushing a foot down on the brake. So when we screened a set of beta blockers, we find that one common beta blocker, metoprolol, it actually causes a decrease in the FRET for the S, which is consistent with it basically shutting down basal signaling through S. But surprisingly, it also increased interaction with the I peptide, which is unusual, has not been characterized before. ICI, which is an arrestin biased agonist, so it should not do anything in this realm, shows no modulation. So this is a specific modulation. Now we've uncovered this using a FRET-based method. Can we test it using something more simple or more well-established? And that is using pertussis toxin. So pertussis toxin ribosylates GI, prevents it from interacting with the receptor. And what you see here is now when you stimulate cyclic AMP production using forskolin and you throw metoprolol, in the presence of pertussis toxin, the effect diminishes, but doesn't happen so for ICI, meaning that the pertussis toxin sensitivity of metoprolol suggests that you are having an effect of metoprolol on GI and also on GS. And this becomes important because metoprolol is a clinically prescribed beta blocker. It's not just important in terms of setting up the adenylate cyclase cyclic AMP set point, but if you activate GI, you now have other effects. The beta gamma subunit of GI goes on to then activate, say, PI3 kinase, you get HERC1/2. So you get multiple effects in cells that are different between these effectors that you now have to consider. And so beyond our lab, once we publish this, multiple groups are now using our sensor platform to sort of profile different ligands and monitor their selection, ligand-dependent selection for different pathways. This is information you want to have so that you don't have quote unquote side effects which are on target but not intended. Another thing you can do with the system is now that we have lots of GPCR structures coming out, if you open Nature or Science, you find another GPCR structure. You can try and interpret what's going on in these structures. So a classic example is we have this ionic lock. It's called an ionic lock based on structure, where you have these interactions that are posited to hold the receptor in a conformation that's not conducive to G protein binding and stimulation. But when you look at, if you mutagenize these residues, you see something that's not intuitive. So if you mutagenize the D residue, you get an increase in signaling, which you would expect if you activated the receptor. You mutagenize R, you basically do not get that increase in signaling. And so this does not help you understand the structure as to whether it's an ionic lock. So we can break the tie by essentially making those mutations and studying them in the context of our sensor. And when we do that, what we find is that either of those mutations increases binding to the G protein. And the reason that this does not activate the G protein, the reason that this does not give rise to an increase in cyclic AMP is because this mutation does not activate the G protein. So we can decouple conformation from function using this method. So this allows you to sort of now look at structure, make some predictions, and look at whether those predictions make sense. Another thing we can do is we can start profiling mutations that are in disease, and now in the context of personalized medicine, more importantly, for single nucleotide polymorphisms that are potentially implicated in disease. So an example of this is using rhodopsin. You have these residues that are mutated in retinitis pigmentosa. What was really nice for us is that when we line up these residues and look at the fret sort of in this sort of map, essentially this lines up exactly with the potency of those mutations in propagating disease. So you can go from sort of modest effects to complete blindness based on the fret profile that we see here. So this gives us another tool to sort of be able to get at the first level conformation information using an assay that's scalable. Yeah.
Allowed previous one.
Yes.
Allowed association but not activation.
Yes. And sorry, this is a different lock. You mean? Okay, let me take a step back. Maybe you're talking about two different locks. Both of them allow the association. There's a last piece of data that I need to start showing, which is this mutation does not promote GTP gamma S cycling. Yeah. Well, that I don't know, except I know that it's not related to the inability of the G protein to bind. I think it's probably more subtle where G protein can bind, but is binding in a more dominant negative fashion, but it's not binding and then being activated potentially the way has been positive. We know for a fact that this mutation does not increase GTP gamma S binding to the unit. Okay, so what's nice is that sort of we've been able to now get some grasp as to how to start looking at conformations of proteins. But the problem is that conformation of GPCRs is fun and interesting and hot as a topic to study, but there's a problem in that conformation has to work inside the cell where the cellular context really matters. And by that I mean that what G protein is available. So in the heart, you have an abundance of GS. In the CNS, you have an abundance of GO. And in the eye, you have an abundance of transducin. So availability has to some extent influence what sort of signaling you will get from that system. And one of the problems is that availability is not always taken into account, even in the context of when people do drug screens and they try and put a receptor on a cell membrane and look at signaling, it's whatever G proteins that the cell provides at that point. So the data and the information that they get is going to be biased by that cell's native G protein expression. So what we wanted to do is we want to take availability out of the picture and then ask the question, in the presence of equal availability using our system, so now instead of taking just the peptide, we take the full G alpha subunit, can we start defining some rules of GPCR G protein interaction? So we've taken a set of different receptors. They signal through different pathways. Name's not important. The main point is when we now profile the specific fret or the interaction between the two of them for the same concentration that we provided using this linker, we find a surprising result. The GS or the G alpha S subunit interacts more strongly than I or Q, regardless of the different receptor types. So the green dot is always higher than the blue or the black dot. And that's interesting. So the GS protein binds with higher avidity. And that would suggest that GS, even though it may not signal, it might sequester a G protein coupled receptor if it's available in excess. And that's what we see. So if we take alpha 1, which signals through G alpha Q, and we look at IP1 signal, which is coming down from alpha 1, and then we overexpress the G alpha S subunit, we basically see the specific FRET decreases, and the IP1 production signal also decreases as a function of G alpha S expression. You can do this in reverse. You can then ask, well, are all GPCRs equal in terms of binding G protein? And the answer is again, no. So not surprisingly, different receptors have different avidities for the same G protein. And then you say, well, if conformation was really the driving factor, then how much of availability you have may or may not dictate the signal that you see. And the answer is it's not correlated at all. So basically, even though you have, say, alpha 1, which interacts with GS with high avidity, you don't see much signaling through it. So conformation does matter. And alpha 2. Oh, good. Sorry. So basically, the units are, you can interpret that as a change in FRET ratio. So if it was a change from 2 to 3, that would be, so 1 to 2 would be 100% change in FRET ratio.
It's a different amount of fluorescence resonance energy transfer.
Yes.
Which means a certain degree of closeness.
Okay, maybe I went into it too soon, too far. Hold on. So basically, no, it's a good question. I kind of maybe skipped over some of the basic details. So basically, the way the system works is you have an acceptor and a donor. And if these two elements interact, you see an increase in FRET. So the FRET change is an indication of the strength of binding between this and that. So the higher the specific FRET, the more binding you have between this and that. So what's nice about our system is this 10 number that's in here. In the basal condition, we don't have much FRET. So the FRET only increases if you have an interaction. So the higher the bar, the more the interaction. So it's a measure of the affinity of the interaction, if you will. So basically, this says that alpha 1 interacts with higher affinity than alpha 2. And this is the sort of profile of affinity of interaction in cells. And then this says that the profile of affinity of interaction does not correlate with the strength of signaling. So conformation does matter. What this also tells us is that if you look at this data, that some GPCRs, for example, this one, alpha 1, which binds with high avidity but does not signal much, shown here from cyclic AMP, can sequester another GPCR. And that's what we see. So for example, beta 2 signals through GS. This is beta 2 GS. You see cyclic AMP levels. If you overexpress alpha 1 in the background, you essentially see a decrease in specific FRET. So the interaction between beta 2 and GS diminishes, and the cyclic AMP levels also decrease. So what these pieces of information say is that you have to have a large grain of caution in interpreting some of the FRET studies that have come out in large numbers in the last several years, meaning that people typically tag one protein with a donor, another protein with an acceptor, and make a measurement inside the cell. Well, the dark matter in the cell, the other proteins that are not being thought of, but can potentially sequester these elements, and also the stoichiometry of the expression and the level of expression can completely alter your interpretation of the outcome. So here's an example of a breadth study where they're basically profiling interactions between different G proteins. This isn't to say it is incorrect. It's just that you see the interaction, but whether that interaction is functionally meaningful, you need to do something else with it. So then it still begs the question, well, how do we then still translate that conformational information, which we know and it's so clean from a structural study, into a physiological context? And what we can do is now we can use our same tool to modulate the effective concentration. So in essence, instead of just using one length of linker, we can vary the length of the linker, go from 10 to 20 to 30. And so what I'm showing you here is a system where the beta 2 has been tethered to the GS, and there's no ligand. There's basal activity from this receptor. This receptor is basally active. And as you increase the length of the linker, the specific decreases, suggesting that the interaction decreases in strength. Because again, the FRET is a measure of how frequently these two are interacting with each other. And what you see quite nicely is that as you dial this interaction, you also, the level of cyclic AMP that you find in the cell follows the interaction quite nicely. Well, you might say, well, I can do that for any receptor. This is just sort of an artifact of how the system is being set up. But it depends on the specific pairing. So beta 2 is known to signal through GS. So you see FRET decrease, cyclic AMP decrease. Beta 2 is not known to signal through GQ, so you can tether it to GQ at varying concentrations. The interaction changes, but you don't see any signaling come out of it. You can do it in reverse. So basically, you can take a receptor that normally signals through GQ, and you can provide it with varying concentrations of the GQ pro component, and you see that the FRET ratio scales, and so does the IP1 levels. Alpha 1 does not interact with GS, although you can tether it with varying concentrations of GS. You can see scaling of the interaction, but not any function. The important thing now is now you have a tool to go in a cellular context and manipulate a single interaction. All the other components in the system are still preserved. So if the alpha 1 receptor wants to interact with an endogenous partner, it can still do that. But I can just modulate this interaction and learn about what's happening in the cell, which you can't do with a traditional approach where you just put two of them and drive expression to whatever level you want. So one direction they're going in, so I just want to highlight the scope of this part of my talk. So one direction they're going in is now we can ask ligand-dependent selection in a cellular context. So for example, here's a receptor, you add a ligand on it, potentially it can populate a set of conformations that activate, say, GQ, and populate a set of conformations that activate GS. And so how do we understand the differential potency for activating these two pathways? One way we can do it, which is more controlled, is we can present it with, say, GQ. So this is phenylephrine for alpha 1. We present it with GQ, it activates GQ. We present it with GS, it activates GS at the expense of GQ. This is already saying that context is hugely important in trying to figure out how to measure the signaling that's happening. But it's more complicated than that. So you have a compound like cetirizolene, which if you present it with GQ, it has about the same level of activation as the phenylephrine, but it doesn't signal as well through GS. However, if you present it with GS, it doesn't signal through GS, but you shunt out the signal through GQ. So for this particular ligand, if GS is present, you would see a dominant negative effect. Whereas for this ligand, if GS is present, you would see a shunt in signaling. So what's nice about this is now, I won't go into the details, but we're coming up with a better defined bias factor, where we can actually take different ligands and profile them in terms of their potency for activating two different pathways that we're trying to modulate differentially through the same G protein coupled receptor. Another thing we can do, and this is a direction my lab is really going into, is we can also couple selection to function. So there have been numerous reports of sort of differential signaling of a receptor or a series of receptors to two different pathways and how that might control a range of different things in the organism. And now we have a tool that we can go in, especially with genome editing, which is starting to modify at the genomic level receptors in terms of a receptor is tethered to a specific G protein and see if the signaling bias is in fact important for specification of a higher order function. So let's say cancer metastasis or behavior are classic readouts. To drugs, in the sense that drugs that would essentially switch the signaling specificity. But right now the assays for those don't exist, and we're trying to create those assays at the level of organisms.
Yes, but could you also develop assays to measure cytotoxicity or some other function other than this? You know, could this be a model system to look at how drugs would interact with a model system to measure cytotoxicity?
If it's off-target effects, probably not. If it's on target effects that are cytotoxic, in other words, if it's hitting the right target, but if it's a side effect of that target, that's precisely what we would hope to measure. Yes. So in the interest of time, I'm going to just talk about only the third segment of my talk, but this is sort of another exciting direction we're going in. And that is now you go one level below. So I've told you about conformation, translating conformation to protein interactions and how to kind of regulate those and learn about selection in a cellular context. And now we have to go to one more layer of complexity, and that is sort of multi-domain proteins. And a classic example of a multi-domain protein that we work with in my lab is protein kinase C. Protein kinase C is one among a class of AGC kinases, about 1 out of 60, shown here in a strip diagram. The conserved domain is the kinase domain. But then you see a range of regulatory domains that are different in different kinases. And that's one way that kinases have evolved to sort of give them higher order function. Another classic feature of AGC kinases is the C-tail domain as well. It's sort of a unique feature of AGC kinases that sort of has a band that wraps itself around the kinase domain and is important for the regulation of kinase activity. So a big feature of kinases is that you have a highly conserved catalytic domain or kinase domain, and that's evolutionary pressure because you have to catalyze phosphorylation. And so the way you can get diversity of function is by putting different types of modular domains, which then can target the protein to different subcellular compartments and potentially specify divergent function. So anytime we think of a modular multi-strip domain protein, the first thing we're likely to do is to use structural homology to kind of bin and silo the different domains. And then based on our information from some other proteins that have been characterized, assign functions. So essentially, this is a pseudo-substrate domain because it looks like a substrate. These domains bind diacylglycerol and 4-ball esters, and so on and so forth. The harder part with multi-domain proteins is the regulation through inter-domain interactions. And those are much more elusive and hard to come by. So in PKC, the classic one is that the regulatory domains can fold back on the catalytic domain through autoinhibition, which is fairly common. And when you activate the protein using small molecules that can bind these regulatory domains, the regulatory domains then swing open and the catalytic domain is left to then go on to phosphorylate proteins. This is just one layer, and even this layer is not completely understood. Often you have higher order interactions, which are even harder to study using any approach that we currently know. And the white whale of the PKC field is dimerization. So people have shown as back as 1999 that if you activate the PKC and through a cross-linker, the activated PKC will dimerize, or you will see at least a dimer band. And also that PKC shows a concentration-dependent change in activity right at about 1 nanomolar. The difficulty is these papers have been published and then nothing really comes out of them. The current model is still that PKC opens up and is active. Part of the reason is because you've got all these domains and it's difficult to map an interface, especially given there's a lot of flexibility. It's unlikely, although I wouldn't say impossible, to say crystallize a unique state, or even using cryo-EM come up with a defined set of conformations that you could then map to say see an oligomer. And I should also point out that these dimers we're finding are also, there are higher levels of interaction, which makes it more challenging to sort of structural biology approaches. So we decided to take a crack at this problem, and I'd just like to give you a few hints of this so that you can kind of see how we use our technique to kind of take this apart. So you can do sort of more traditional biochemistry approach where you basically look at the protein-protein interactions. In this case, because we do a lot of FRET, we can basically see if when you activate the protein, so you go from active versus inactive, you see an increase in FRET, that means that there's an enhanced interaction between these two proteins. And so in the wild-type protein, you see that interaction that's been also reported using other methods. Now you can start doing the classic biochemistry trick, start lopping off domains. You take off the C1 domain, interaction goes away. Take off the C-tail domain, interaction goes away. Take off the C2 domain, interaction partially goes away. And that's a problem because that means that the interface is not just one spot, but there are multiple points of contact. You can do another trick, which biochemists often use. You sort of group the domains and start throwing them in solution and see if they interact. So you ask if the regulatory domain interacts with the catalytic domain. The answer is, okay, when upon activation, the two regulatory domains interact with each other. If anything, the regulatory catalytic domain interaction is slightly weaker, if not changed, and the catalytic domains don't interact with each other. And this would be problematic because what it would suggest is basically that you have a nucleation event that these two interact with each other, but then there's data that take away from that, that if you take away the C-tail domain, which is this band right here, you lose the interaction. So I'm showing that here again. Now again, if you put fluorophores at the N termini, this shows an increase in interaction. But if I just take away one of the C-tails, I completely lose that interaction. So this is where you would typically get stumped with a sort of traditional biochemistry approach because you have no more insight into the interaction. So what we do is we now put this linker element in between domains. So we can just take this linker and drop it between domains. And what this allows you to do is allows you to modify one interaction while keeping all the domains. It's not surprising that when you start taking domains off, especially when you have a multi-domain interaction, that there's going to be cooperativity or synergy between those interactions. So here, if you were to put one fluorophore here, one fluorophore here, weaken this interaction, and ask, does this part interact with that part? The answer is yes. I put these two linkers in between. Does this part interact with this part? The answer is yes. I'm only showing you the positive data. We have multiple controls to show that this is not non-specific. The idea then is that you have a nucleation event, but the nucleation event is not sufficient to sustain the interaction. You then have other interactions which then cause the protein to fold upon itself and form a dimer. Well, this is interesting. You can do this in a different way, which is much simpler, where you take this modular domain, which has a donor and acceptor with a flanked linker, and start moving it through the protein. And essentially, if you hit domains where things on the left interact with things on the right, you will see an increase in threat. So this domain interacts with this domain, check that, you see an increase in threat. This sets of domains interact with this domain, you see an increase in threat. That pseudosubstrate domain does not play a part in this interaction, and so you don't see a change in threat. And what I'd like to leave you with, in the interest of time, is the idea that instead of trying to take a traditional approach, and this, I could substitute this for saying, take antibody A, pull down A, and look for an interaction with B. This is just threat here. So instead of using a traditional bimolecular approach to dissect an interface, just start using this system where basically the readouts that you see here. So in this context, again, if I got rid of the C2 domain, I lose partial interaction, much similar to this. If I got rid of the C-tail domain or a portion of the C-tail domain, I lose this interaction. So they both give you parallel readouts in terms of mapping of the interface. It's just that it's much simpler to some extent. I'll just make a couple more points. One is that I showed you a lot of data using FRET. We can confirm that using cross-linking. So even if you come across this and you say, well, I'm not sure the FRET changes are difficult for me to interpret. Well, an example would be in this band domain here that I get rid of, there's only a single cysteine. So if you have a cysteine cross-linker, I completely get rid of the dimer band. And that kind of confirms orthogonally the interface we've mapped. And function, does this matter inside the cell? And we think it does. The reason we think the PKC dimerizes is because of an equilibrium mass action argument. So you start with a non-active state, you go to an active state, you're likely to just come right back. Whereas if you have a dimer, the interface latches close to these same interfaces that would then shunt you back. And so you have a little bit of activation giving rise to a lot of activity in PKC. And we can test this inside cells. So because we know the interface and the domains, we can make predictions, and we can take peptides from these interfaces. Here's an example of a peptide that if I made it into the native sequence, I can block the interaction as seen with FRET. I can block it in live cells. And more importantly, I can block the interaction without driving the PKC off the membrane. So even though the PKC is activated, it goes to the membrane, and that's sort of the hallmark of how you think of PKC. You think PKC on the membrane equals PKC active. And what we find is that even though the PKC is not driven off the membrane, when we throw these peptides on cells, you basically completely abolish a classic PKC downstream response, which is ERK1/2 phosphorylation. So basically, what you see here is the PMA you get a nice increase in ERK1/2 phosphorylation. If you throw this peptide metastylated on cells, you completely block the response. Same is true for LPA, which is now driving through an endogenous GI-GQ signaling pathway. You can completely block the response. And this response is specific to PKC. If you throw something non-characterized like serum, which is going to activate ERK1 through multiple pathways, you block the response only to the extent of a PKC inhibitor. And the main advantage of using the system now is you now have a way of targeting PKC function, and more importantly, distinguishing between different PKC isoforms in a way that you would not be able to do using the active site, which is highly conserved between different kinases. And the last two slides I have promised is the first one is, while we were doing a lot of these studies, we found something interesting. And that is, when you start playing with these sorts of sensors, if you put something inside the active site, so now I'm putting the inhibitor BIM1, we find that not surprisingly, the conformational changes in the catalytic domain are relayed outside the active site. So if you put the inhibitor into the catalytic domain, you weaken this interaction between the regulatory domain and the catalytic domain, and that is shown here. And if you weaken this interaction by mass action argument, you actually stabilize this state. And that's what you see. You get more dimer being formed. And that translates into a non-intuitive result for PKC, which is that if you now throw an inhibitor on cells, so here's a classic PKC response, PKC into cytosol, activated, goes to the membrane, comes back to the cytosol. Sort of a transient response that you would track in response to GQ activation. So if you throw BIM1 on cells, you actually stabilize membrane localization of PKC, which is completely non-intuitive. You have a PKC inhibitor that prevents PKC function, stabilizing membrane localization. And the main point that this is arguing for is for moving away from thinking of these sorts of enzymes that are multi-domain as sort of on-off switches that you can simply manipulate from the catalytic site, but more towards sort of using these tools to understand the allostatic modulation of kinases. Last data slide is this one, which is just to show that we can now throw different inhibitors and map these interactions. A good example is inhibitors that don't distinguish between alpha and beta of PKC look very similar. But riboxystarin, which is a putative PKC beta 2 specific inhibitor, we can distinguish between PKC alpha and PKC beta 2. So it shows a beta response in beta, but not in alpha. And last point I'll make for the talk is we're using saturating concentrations of inhibitors. So at saturating concentrations of inhibitors, you would, in a traditional kinase assay, not see any difference between the two isoforms, but we're able to detect small changes in there that we would otherwise not detect using a traditional kinase assay. So in the interest of time, I'm going to skip across to the acknowledgements. These are the people who did the work. I didn't have a chance to talk about our work on myosins, and that's being spearheaded by Rizal. Ruth is the one who's sort of working on both myosins as well as PKC. Rabia is spearheading our efforts with GPCRs. I don't have a picture yet for Ansley, but Ansley's also joined the fight on GPCRs recently. Carter's the one who's spearheading all of our thinking on allostatic conformational changes in PKC. And Mike sort of plugs into most of the projects that I talked about right now. With that, I'd like to acknowledge funding sources. Thank you for taking the time to listen to us.
Okay, thank you. I'll invite questions. Maybe I'll start with one, Gil. So this is a very elaborate allosteric regulation mechanism that you've uncovered here with the C-terminal tail. Is it unlikely in your mind that this is unique to PKC? You showed that big kinase or little snapshot of the kinase superfamily. Do you think it's happening there? It's been elaborated.
I'm looking at Ruth because she has an answer, but I'll give the answer on her behalf. So we've tried AKT and S6 kinase 1. We see different types of modulation, but we do see modulation. So it's not unique to PKC. And also there was a recent report for focal adhesion kinase, and we've been able to reproduce it in our lab, that there's a firm domain FAC kinase inhibition, and that's also sensitive to nucleotide. The difficulty right now is in our sensors, it's sort of a binary readout. So either it increases or it decreases. So the challenge for us is using a more traditional approach like say NMR to then understand get some high resolution information as to what those conformations are. Our goal ultimately is to be able to hopefully silo some of these inhibitors based on the conformational changes they have. Because the benefit of the technology we have is we have the resolution certainly in terms of the dynamic range to, along with some tricks of our own, to kind of see if we can silo inhibitors in a primary screen rather than just look at them as in terms of their absolute effect on activity.
But the CTL itself isn't conserved across the superfamily.
It's not conserved in the sense that we see it with kinases that have different CTLs, for sure. The CTL effect, well, if you take off the CTL, the effect goes away. So that is part of it. But the CTL, yeah, you see this with kinases that have different CTLs. Gil?
First of all, congratulations on the Biological Sciences Scholars Program that brought you here and for listing it. And I'm glad the group is getting together in November. I'm interested, besides the point mutations and the conformers that you mentioned, do you have evidence that there are splice isoforms of the G-coupled protein receptors or the proteins that are in the pathway?
So we haven't played in that space yet, but I suspect that is going to be the case. So the receptor that we've looked at that's closest to your question is the alpha-1 receptor, and there are multiple different splice variants. And there's reports in the literature, as probably where you're going with this, is that those splice variants, because of changes in either intracellular loops or CTL, might be signaling differently in terms of their profile. That's a great suggestion. We haven't explored that, but I would suspect that those will have an impact in being able to differentiate between the functions of those receptor isoforms.
So you mentioned the concern that expression levels, especially binary reporters, could have effects on the measured results. You didn't tell us very much about the expression levels of the monomeric fusion proteins that you're studying. I guess the same issue comes up because they're certainly part of networks of interactors, other proteins that they function in concert with, beta gamma complexes, other co-receptors. You also mentioned the context dependence, and I was wondering, are you doing this in primary cardiomyocytes, or what is the experimental system where you're testing these effects?
So actually, it's a great point. So I wasn't meaning to imply that our method is substantially improved compared to what has been published in literature. I guess my main point was that people generally tend to transiently transfect two components and look for changes and how that flows. I think our data primarily allow for comparison. So one important caveat is all of our comparisons that I showed you are done at equal level of expression across all systems. So when I make a comparison, I am taking the trouble to make sure that the expression level for two different, like if I'm comparing alpha-1 with alpha-2, alpha-1 with beta-2, that I am making that comparison. So we have been primarily doing work with HEK cells. We have systems to stably express, but most of it right now is using transient expression. But that is part of the limitation right now, is that we definitely want to control for expression between experiments and between conditions. One benefit the system does have is they're both expressed through the same, as part of the same protein, so at least the stoichiometry is not a factor. But the absolute expression will determine some of the outcomes because of all the background effects that you're talking about. So to avoid that, we basically control for expression, experiment to experiment. So essentially, per cell, the average fluorescent protein count is going to be the same. But that is a limitation in terms of being able to scale up. We have some methods of doing it through either viral or stable lines, but they haven't been perfected yet. But hopefully when those get perfected, then it won't be, I mean, I can be able to do this instead of maybe a high throughput format. That would be the ideal.
How evolutionarily conserved are the G alpha S, G alpha Q, G alpha I in terms of the region that's interacting with the GPCR in terms of sequence and then also the structure?
Yeah, so actually, the reason I'm able to do experiments between Si and Q is because they're very different. Within the family, so let's say GQ versus G11, G alpha I1, 2, 3, they're very similar. So I don't think our system will be able to differentiate between those three, but big differences, there are huge differences between S and I, both in terms of the C-terminal peptide as well as the interface that the G alpha protein presents to the receptor. So they're very different. There is hardly any sequence conservation. In, for example, the C-tail is there's the alpha 5 helix that comes out. There's some residues that are similar, but in the very C-tail element, the sequences are completely different. Okay. Between Si and Q, but between sub I versus O, I1, I2, I3, you can silo it into I subtype, S subtype, and Q11.
Okay, and then so then the those other subtypes, I guess, are they like in terms of the downstream effects? Like how does that correlate at all with the level of
interaction between... yeah, okay, so that's a good question. Once you start getting into G alpha I subtypes, the main data that would suggest differential function will be from, say, knocking out a particular subtype, saying that that subtype is in fact important. So that's where the data would come from. From a biochemical assay, you won't see very, very many differences. So it might be simply what subtypes are expressed in what cell types, and potentially post-translation modifications or where those subtypes go would determine the function. So it wouldn't be, I wouldn't expect it to be at the level of this modulation. If that is maybe related to your question.
Yeah, okay. Thanks.
So when you test your different binding ligand, do you know the binding site, or are different ligands they bind to the same site or different binding site?
So these are all orthostatic binding ligands that we've been working with. We're not looking at anything that's allostatic.
You don't care?
Well, no, if you are trying to study two ligands at the same time, then we care if they're occupying the same site or not. If that's where you're going.
Because you know the structure, right?
Yes.
Here, so you can probably do a kind of binding site search, and then that can guide you to select your ligand.
It's possible. I think that comes back to the question earlier, which is our technique refined to the point that we can actually start screening? The answer is not quite there yet. But it could be a way to do it.
So currently, how did you select the ligand you test?
Basically, we know, there's libraries of ligands that are already characterized, established. People use them in different assays. So we're working right now with ligands that have already been tested and characterized. So we know at least that these are specific to this particular receptor subtype.
Okay.
So it's quite striking that GS has higher affinity for all the guys you looked at. Any idea why? Any of the properties of GS? That is it, electrostatics to the membrane? What could it be?
So one thing I
think that's new data, right? I haven't.
See what? That's new data that's completely surprised us. Anecdotally, the closest I can, the only thing I can tell you is it's not related to nucleotide cycling. So in other words, if we make nucleotide mutants, because the different G protein subtypes cycle nucleotides at very different rates. So we can make nucleotide mutants of the different family members, and you wouldn't switch based on nucleotide cycling. So that partly answers it that it's not related to cycling. But we don't know. What I didn't present is negative data for that experiment I showed you where you would ramp up GS and see a shutdown of signaling. We can take another G protein subtype that doesn't bind as avidly and overexpress it, and it wouldn't do the same thing. So there is functional data in a biomolecular sense that there is something to what we're seeing with our FRET reporters. What the reason for it is, I don't know. Interesting. That's an interesting point. I mean, I think for beta 2 and GS, we know the interface. Beta 2 GI, hopefully we'll have one in the coming years. Because we only have a co-crystal structure right now for the GS. We don't have a co-crystal structure for GI. Theoretically, we could just dock it in and look to see if interaction. Okay, that's a possibility. Yeah.
Okay. Well, if there's no more questions, let's thank Shiv again. Very nice talk. Thank you.